All right, welcome everybody. Thank you so much for being here today. I know it is not only a beautiful day outside on this very sunny fall day, but there's also lots of activities going on in the world, and you chose to be here. So thank you for being here today. We've got some really great presenters who are going to be sharing the latest about ALS research. And I know that I don't have to tell the folks who are sitting in this room what it takes to do ALS research, how much it costs, how much time it takes. And I know many of us are often disappointed when clinical trials or research don't meet their endpoints. And I think a takeaway that we'll hear a lot about from the presenters today is that it takes time, and sometimes those things that we deem as failures are actually teaching us how to do research moving forward, and that we have a lot that we can learn from things that go wrong. So today, I'm just really excited to share some of the presenters that we're going to be talking with. And this year, we're doing things a little bit differently. For those who have come to this event in the past, we've typically spent the majority of the afternoon having the opportunity to hear just from the speakers. And we will be doing that this afternoon. But the second half of the afternoon, we're actually going to have our presenters at the back of the room. And we really encourage folks to walk around and talk individually with those individuals and learn a little bit about opportunities to get involved in research, and learn a little bit more about the science. And a lot of that's happening right here in our backyards in the Pacific Northwest. I do want to just say a couple of quick shout outs. Thank you to the Middleman Jewish Community Center, who is hosting us, I think, for the probably 10th time in a row now. So thank you to them. Thank you to Versa Events, who is helping with all of our technical needs this afternoon. And thank you to our sponsors, MT Pharma and United Access. And of course, thank you to each and every one of you for being here today. So without further ado, I am going to introduce our first speaker of the day, Dr. Nicholas Olney, who is the medical director of the Providence ALS Center. Nick is going to be giving us an overview of clinical trials and all that's happening over at Providence. Dr. Olney received his medical degree at the University of California in San Francisco. He completed his neurology residency at the University of California in Los Angeles. Following his residency, Dr. Olney returned to UCSF to focus on neurodegenerative disease and research prior to his joining the Providence ALS Center here in Portland around 5 or 6 years ago. Actually, a little further than that now, huh? 7 or 8. All right, well, help me in welcoming Dr. Olney to the stage.
Oh yeah, thank you. Thank you so much for inviting me. Very happy to be the opening act here for this exciting research symposium. So I'm the director of the Providence ALS Center and Clinical Trials. This picture in the background actually is from Black Butte, which was my dad's favorite place to vacation, both before he had ALS and when he had ALS. And actually Lance, who is the president here, who just recently retired, he actually delivered us a lot of the adaptive equipment to make our cabin so we could actually handicap-proof our cabin, which was great. So I thought when I saw him on my first day interviewing here that Providence would be a good match for me. So this is a picture of my dad. Oops, is the slides down here advancing, but there's a. Sorry, they're off a little bit. Okay, there we go. We're synced. All right, sorry about that, little off sync. So this is a picture of my dad recently after he was diagnosed with ALS. Here's a picture of me as a caregiver with my dad as a family. But I can say that seeing how devastating the symptoms of ALS were firsthand is what made me want to learn as much as I could about this disease throughout my training. It ultimately led to me wanting to join Providence and be part of clinical trials so I could be part of the winning team to find effective treatments against ALS. So to start off, I'll just tell you a little bit of clinical trials 101. So we have 3 phases of clinical trials. Phase 1 is typically the first time they're ever in humans. Typically that's about 10 to 20 patients. typically those aren't coming to the Northwest. Traditional phase 2 trials will be different dosages and everyone's getting drugs. This is where you pull some of the early clinical data as well as safety profiles and what's the right kind of sweet spot to give the dose for future trials. And then phase 3 is really that trial that the FDA requires to prove that the drug works better than not taking the drug. So patients get randomized to either drug or placebo or the sugar pill. And I think the ALS space has gotten very creative with different kind of clinical trials. So I think one of the biggest and most popular one at our site has been the Healy ALS platform trial. This one, as compared to traditional trials, so traditional trial, we have to go through all the regulatory, build a big stadium, run the race or the study, and then tear it down, and then you analyze data for the next months to year. Here, the platform trial, we built a very big stadium that can run multiple races at the same time, and it stays perpetually open. So as one race ends, we can start other races, and that's kind of the place we're right now. Like, we don't have any active recruiting regimens, but we hope we will soon, which I'll tell you about in a second. Some of this is driven by big statistics that we can actually pool the placebo, as you can see here, and that allows patients to get 3 out of 4 chance of getting active drug. This also can cut the time by a third, or sorry, cut costs by a third and cut time in half to getting discoveries. We collect a lot of the basic endpoints, so ALS functional rating scale, breathing with slow vital capacity, the hand grip, survival. There's specific biomarkers for each regimen. Those are almost becoming required now. They also have a great way of importing in exploratory endpoints and safety endpoints. We were one of the first sites to open in 2020. This is a map of all the sites that are currently open. I think it's almost doubled since we first started. In the last 5 years, since 2020, we've gone through 7 regiments, all with different targets. And I think we did learn a lot, as Cassie was mentioning earlier. Even the ones that aren't successful, we know like these are the targets we should go after. These are ones we probably don't want to go after as much anymore. Two of the regimens, so regimen C and D, are going on to phase 3 trials and are planning phase 3 and had expanded access programs. So that would be regimen C, the CNM-AU8, had a survival signal in the Healy, as well as a small trial in Australia. So they need to have a bigger trial for it to be FDA approved, but I'm a fan of that drug, and we are a part of the expanded access program. The regimen D, prodopidine, was a sigma-1 receptor agonist and had a signal of benefit in definite and early ALS, as well as some slowing of bulbar function in the exploratory endpoints. And then Healy, actually just a couple weeks ago, has announced 3 more regimens that are coming soon. I know the most about the first 2, so I'll tell you briefly about those. There's a Lilly oral molecule which inhibits SARM1. It's predicted to help stress or injured neurons. And then Neurizin has a molecule focusing on autophagy. It's NUZ001. The guy was from Australia who presented that one. It's focusing on the pathway of autophagy that's supposed to help eliminate toxins as well as potentially TDP-43. And so we're hoping that the Lilly one might be available by the end of 2025, but definitely hopefully in early 2026. There has been some changes to the Healy protocol in that rather than 6 months of patients getting definite drug, once they enroll, it's going to be more like 9 months. This is kind of just learning from the prior studies. Also, the inclusion criteria, rather than being 3 months, is now 2 years. They're also focusing on more remote visits, as well as biomarkers, including PBMCs for stem cells. We also are big into the expanded access programs. Anytime we have one, we try to get involved. This is for patients that do not typically qualify for typical clinical trials, but patients get definite drug, no placebo. Also, it's a way for patients to get access to drug and also to get more safety data on these drugs. So we're involved in the Clean Nanomedicine CNM-AU8 expanded access, as well as this last year we got into the very exciting RAPA cell-based therapeutics into the TRIG cells, which I talked quite a bit about last year. And both these are fully enrolled. They enrolled actually pretty quick. And so hopefully have some readout on these next year. We're also part of multiple ALS registries or consortiums for patients that aren't able to do clinical trials. So All ALS, which is my favorite one, especially since we have our boss, Bob, here in the audience. We also have the ALS Natural History Study and CREATE. We're also still looking for ALS patients from other centers, also anybody with family history of ALS or controls. So I try to get a lot of my friends and family involved. Anybody that asks, what can I do to help with ALS research? I say, volunteer for All ALS. It's great. So we'll have a lot of flyers in the back. We also have the website, and that's the email of my lead coordinator for this study. And then in the last couple of minutes, I want to tell you about one of my favorite stories, I think, to come. I've actually been following this technology for probably the last 20 years, but the ALS genetic treatments. So this is only for the SOD1 population, which is only about 2% of overall ALS. But we have mounting evidence that SOD1 is a toxic protein. So to go over one of the dogmas of earlier cell biology, we have the DNA that makes RNA, and then that will translate to the protein. So if we know that the protein is bad, is there a way that we can block that? And so that's where we bring in this mirror image RNA, or a antisense oligonucleotide, or ASO, that will block that so that the protein is never made. So with that actually was the premise. It took many years of studying this, but the VALOR study, which we had patients enrolled in, was launched. It is an intrathecal delivery, so you have to give the drug through a lumbar puncture. The results of this study came out in a New England Journal of Medicine paper, which I'll go over briefly here. So here we have red is getting the drug early, and blue is getting it later. And so here we have a graph that after basically 12 weeks, both of the groups show the SOD1 protein concentration going down in the spinal fluid. We also see here neurofilament going down at around 12 weeks, and neurofilament is a biomarker for kind of neuronal death. So if that's going down and staying down, that's a good thing. And so I think both of these slides show good target engagement for this molecule, the tofersen. Then we start looking at the clinical endpoints. We have the ALS Functional Rating Scale. So it did not show clinical significance at 28 weeks here, which is that middle line. But if you can see, when everybody's getting drug, we start to see more stabilization. Also, we look at other clinical measures like the breathing and the slow vital capacity. We see a similar trend. We see a similar trend here in the hand grip, which is also pretty amazing. And then we also see a signal in the survival benefit, where the red, who got it earlier, are living longer. The most common side effects are procedural and headache, mostly due to lumbar puncture. There are some further rare side effects like myelitis, radiculopathy, papilledema. We're still tracking for side effects since this drug is rather new, but I think most people are willing to tolerate the side effects unless they're rather severe. And this has led to the FDA approval of Quesaldi, which is also tofersen, needing monthly intrathecal injections based on this good data and target engagement and the lower of the neurofilament. And also I was very excited that we were one of the first places to give clinical drug and made the local news, thanks to our NP, Lauren Brown. I've previously shown the video in prior studies, if you want to look at that, or prior symposiums. And then I think one other very exciting part of this study is that just came out. I saw a poster at NEALS, our national ALS meeting 2 years ago, but this paper is finally out that was written by Sean Smith and out of Tim Miller's group. So he's a neuro rehab specialist out of WashU, and he actually has shown a case series of the patients treated with tofersen having stabilization. So looking at this paper, he has patients treated for anywhere between 5 and 30 months on tofersen. So there are different time points here for where they're measuring. They do show the neurofilament and all of them going down, which is a good target engagement again. And then here's really kind of some of the rehab data. So they're showing the ALS-FRS. The blue here on the left on the very top graph is the first ALS-FRS score, and then the orange is the last one, but that could be a different time point, and green is the predicted. So here I think we are seeing some further stabilization of the ALS-FRSs. We're also seeing a similar trend in the muscle strength for the 5 patients, or that's 6 patients. And then we're also seeing this stabilization in the functional independence measure scale, which is a rehab interview that they do. And then this is, I think, the very exciting data, although I have to be guarded about this, but there's even some improvement here in the 10-meter walk test and the sit to stand test. And then the other one, the TUG, is the time up and go, which is a little bit more variable, but those 2 other ones I think are very promising. And so I look at these and I am very hopeful. I'm very guarded because this is a small set of a very small rare set, but I think this is a very hopeful thing that at least a small group of ALS is responding to these treatments, that we have the relatively well understood mechanism. So I'm hoping that if we can, carry this these targeted mechanisms into if we have these targeted mechanisms and spread that into sporadic ALS, that will make significant difference. And I did have a little thank you slide with some people. If we could show the last one with the pictures. Oh, there we go. Yeah. Yep. So I do hope that we can make our ALS centers of excellence into ALS treatment centers in my lifetime. And I did want to say that this is my buddy Judy and Al at Powerland down in Salem at the Great Steam Up, and they always come to all my research talks. So I want to give them a shout out. So thank you so much for your attention, everybody.
Thank you so much, Dr. Olney. That was a really great talk, and I think we're all feeling hopeful seeing those new data points, so thank you for sharing that. All right, I am excited to welcome up, new to our symposium this year, Jack Silva. And Jack is a passionate leader and advocate in the ALS community. He serves as a founding board member of Geo ALS, which uses advanced mapping technology to transform geographic data into powerful tools that improve patient care and speed up research efforts. One of his flagship tools is the ALS Clinic Advisor, which helps patients locate nearby clinics and read reviews from other patients. Their other project enables researchers to analyze data patterns and patient histories in new ways. Jack also co-chairs the Legislative Affairs team at AM ALS, where he helps drive legislative efforts to improve access to treatments and increase research funding. I'm really excited to welcome Jack up to the stage. Let's give him a hand. Absolutely. And just to advance your
Cool.
slides, you can push here.
Cool. Awesome. Thanks. There we go. All righty. How's everyone doing today? Thank you, Cassie. Really excited to be here. My name is Jack Silva, as she said. Been doing advocacy for the past 5 years in what one of my favorite ALS leaders, Sandy Morris, called ALS land. And recently, I've been a part of Geo ALS, and we've been doing exciting work with mapping technology. When the founder of Geo ALS, who wishes he could be here today, was talking about mapping technology and how it could transform advocacy and how it could transform like how we actually strategize and think about clinics and think about our care. I kind of did an eye roll because I really didn't even know what mapping technology was, even though I work in Silicon Valley. And then he said, well, yeah, it could pave the way for even new research and accelerate research. And then I did another eye roll. The irony is I'm here today speaking on behalf of it because I've become a champion of it and in his tutorship. So go to the. Oh. So yeah, I'm gonna get into how he proved me wrong in a little bit. But first, I want to talk about how we're moving the needle on ALS. I'm not sure if anyone of you knows this big, beautiful being right here. His name is Pat Dolan. He worked at a company called Esri for 20 years. Esri builds mapping tools and they build location intelligence. It's powered by a very cool technology called GIS, which stands for Geographic Information Systems. Geographic Information Systems, or GIS, turns raw data into interactive mapping tools, which help us discern and understand patterns of care and other kind of like clinical formats where we could, you know, as an ALS patient or caregiver, address. I'm going to get back to that. Pat here was diagnosed with ALS in 2016. We all know ALS robs you of your ability to eat and speak and move. Up until 5 years ago, or Pat has no longer been able to breathe on his own. However, Pat actually created all these slides today that you're going to see using eye gaze technology. He's doing it from his bed in Riverside in Southern California. I know I'm going to be talking a lot about Pat today. He's a hero of mine, and he's the one behind all of this. So after Pat's diagnosis, he wanted to apply all of the GIS skills to advance the science of ALS. So we teamed up with mapping professionals, researchers, and advocates to help him do it. And we're called the Mappers, and the organization is called Geo ALS. Our mission is very simple. It's to advance the science of ALS through mapping technology. So when I said this technology turns raw data into interactive maps that helps you understand relationships and patterns of the data, for us that means finding gaps in patient care. For us that means trying to identify areas of research that could help impact the ALS community. For us that means helping find where the most ALS clinics are concentrated. Now, when we find that, it opens a lot of doors. We can provide researchers with analysis on specific ALS locations. We empower advocates to educate Congress on what's going on in ALS land. And we allow the community to find clinics nearby them and interact with those clinics so they could see if the needs cater to their individual specific needs. So how many of you are from Portland? A lot of people. Nice. How many of you? So a lot of you drove here, I'm assuming. Who lives the furthest from Portland? What was your longest drive? Everyone's looking around. Hour, 2 hours. So for Pat Dolan, we had a conference in San Diego. He lives 2 hours inland from San Diego. When we think about planning a trip for him to go there, which he eventually was not able to attend, it's like planning a trip around the world. Planning a trip for anyone with ALS is very exhaustive. It's frustrating, and it takes a lot of effort. So even though traveling is hard for people with ALS, going to clinic is really important, especially in the early to middle stages when you're talking about, you're seeing your respiratory therapist, your occupational therapist, your physical therapist. So it's important that people with ALS are able to access the clinics that are closest to them. So our first initiative was to map the ALS MND clinics worldwide. To do this, we created a pretty vast partner network where we had to try to find partners that manage clinical data and that would actually give us access to it. That includes the International Alliance, ALS MND, the VA, as well as the ALS community as a whole, and hopefully soon we'll be excited to partner with ALS Northwest. By doing so, we created the first ever ALS clinic spatial database. And then we also had developed a special anonymous review capability to give people with ALS and their caregivers be able to share that experience with others in an anonymous way, just to kind of give more transparency in the process. And again, the goal is to build the transparency and trust within clinics so more people want to attend clinics and get the best out of their clinic experience. The ALS clinic survey is one of the components of that. It's not a generic survey. It's very tailored. It was created by people with ALS and their caregivers. And in order to do that, we also, enabled some of the neurologists and some of the other clinic functions to come and contribute to that survey. And when they go and contribute to that survey, what's that? Yes, that was intentional, but thank you for noticing. And after that, we added all of these capabilities together in one place. So we created one unified solution called the ALS Clinic Advisor. So the ALS community, the goal is that they could find clinics closest to them, and most importantly, are the best representative of their specific individual needs. So the final part of that is to keep gathering more information and data. We ask them to leave a review on their clinic experience. The reviews we've gotten are very accumulative and very robust, and they're also very positive as a whole. We found that the people living with ALS and their caregivers, Even if they have some constructive criticism, they usually have a generally positive experience, and they just want to make that experience a lot better, or a little bit better. The major organizations that are hosting us on this platform right now are the VA, the International Alliance, Your ALS Guide, AM ALS, and the CDC. And again, we're especially excited to start working with ALS United and hopefully ALS Northwest with their great community here. Now, I'll turn our heads to advocacy. As many of us painfully know, ALS has been around for 150 years, but without any significant drugs out there that have been approved by the FDA. It's still severely under-resourced, underfunded, and it's killing thousands each year. It's killing our veterans, but there's hope. 4 years ago, the Act for ALS passed. It was the first ever government-funded program, and I stress that, the first ever government-funded program for any disease, and it gives people with ALS outside of the stringent requirements of clinical trials an option to access really promising or experimental treatments, and gives the person living with ALS some more autonomy in their decision making to see, hey, let me take a chance on this. It's huge too, because as stressing again, as we're all again painfully aware, people with ALS, most people with ALS are not qualified for clinical trials. The other cool thing about the Act for ALS, it was passed by just a relentless grassroots effort. People like me and you and everyone in this room were petitioning and asking their legislators to co-sponsor it, which was really great. However, the bill is set to expire next year, and if it's not reauthorized, the access disappears. It's cut and dry just like that. So we're kicking off advocacy efforts right now. The first major step is to convince more members of Congress to formally support our cause by joining the ALS caucus. It's no small task. It takes calls, it takes emails, it takes virtual visits, it takes in-person appointments. It takes going up to DC, it takes meeting with your local district rep. So enter a tool that's been essential to this work. We created the Legislative Viewer. It's what this is called. It's in honor of Glenn Rouse, who recently was a part of our group that passed away last year, a great mapper himself. So advocates can identify their reps and senators and see where they stand, whether they supported Act for ALS last time, if they're on a key committee that makes health decisions or appropriations decisions that will fund a bill like this, if they're a veteran. So all of these maps have this kind of intuitive interface that allows the regular person like me that doesn't have a mapping brain to go in and access this information to empower you to have these really powerful conversations with your senators and reps and say, hey, this is very important for the community. We want you to sponsor this. It shows how the ALS is impacting people in their state. So it includes a veteran population score that has veterans have a higher risk, as we all know, of getting ALS, the number of ALS clinics in the area, the number of VA ALS clinics. It also has ALS grant funding to show the impact that your specific district has on ALS or has not had on ALS. So by making this info available, you're turning your advocacy into meaningful action. You're showing up to meetings with a Congress member and saying, hey, Senator, this is what matters to your constituency. This is what you've done in the past, and we're grateful for it. Or we're a little leery right now because maybe you haven't given all of your efforts to something so bipartisan and bicameral like ALS. And then we have the power to say, by using these maps, this is what we're asking you for right now. And it's really powerful too. As Cassie mentioned, I lead the legislative affairs team at AM ALS. Use this map before every congressional meeting we have, before every email that I send. That's pretty much it. we got two things going. We're really excited about a new release coming for the dashboard for our ALS Clinic Advisor. Again, it shows reviews and experiences of past clinic visits from all your peers, so you can kind of get an understanding of how the neurologists are or how your respiratory therapist interacts, so you could kind of have a knowledgeable conversation with them. Again, the goal is to elevate the standard of care in ALS. Questions? Thank you, everyone.
Thank you so much, Jack. It's really interesting to see how our mapping technology is helping not only with advocacy, but also in access to ALS clinics and, of course, to research. So thank you for that. I'm really excited to bring up an old friend, Dr. Joe Beckman. Many of you who've been attending our research symposium in years past have heard Dr. Beckman speak about his research. While he is now retired from Oregon State University, he has been at this work for over 50 years. And he is a man who needs very little introduction. I'm just really excited that he's here today. So let's give him a very warm welcome.
Great. Well, excuse me. Thank you all for being here, and it's great to come up. It's a nice 2-hour drive up. And I'd like to tell you a little bit about some of the things that we're doing. I've retired, but that really means I've retired from teaching, from faculty meetings, and writing grants, but able to focus a lot on ALS work and still go into the lab. I wanted to acknowledge Alex Maharjan, who's sitting over here, who was an undergrad working in the lab for a bit. He's now working with patients here, and I'm going to show you a little bit of data that he collected. So I thought I'd start today by just thinking of what it was like when you were diagnosed and you start to search the web and find out what treatments there are. And it's really kind of scary when you realize there really aren't treatments. There's 3 that have been approved. The first was riluzole. And you're trying to understand this horrible disease. And if you look at the figure on that side, the blue lines are axons of motor neurons. They're basically describing 180,000 neurons that control all your movements, and this disease is causing those to die. So we've been working at this for 30 years, and what are the problems and challenges? And it turns out it's an enormously difficult problem. So riluzole was the first drug. It's thought to work on glutamate, although it probably has many other actions, extends survival by 3 months. The second is edaravone, which is a drug that came from Japan, and it's called a free radical scavenger, and it's known as Radicava. And when it first came out, it was suggested to be, to maybe it only work in 7% of patients. I'm going to come back to that. And then we just heard a talk about tolversen, which a lot of people are excited about, but it really only targets 2% of cases. And what was unique about it is it was approved based on a biomarker. And a biomarker for ALS was a huge step forward. So what I'd like to do today is come back and talk about edaravone. Let's see if, did that advance? There we go. So one of the problems is a free radical scavenger. There are a lot of free radical scavengers in the body naturally, and they're actually present in much higher concentrations than edaravone. So as a biochemist, I'm trying to figure out how in the hell can it work? And the second is that actually there's a lot more data that's coming out that's saying this might work better than you think. So what I want to do today is talk about a potential biomarker that may be related to edaravone and showing that there is a lot of actually great progress. I think we can start to make further understanding of it. So let's see. Okay, there is a delay here, so it's a little bit. So the first plot I'll show was done when this drug was given by intravenous administration, And what they did is follow patients who continued. So the way a drug is tested is usually you try to get a signal in 6 months and then decide to go forward. But as they followed this out, Brooks published this paper that came out 2 years ago that edaravone seemed to extend life perhaps as much as 6 months beyond treating with riluzole, which is actually fairly significant. And then the next study came out this summer. Okay, and there's even stronger evidence that there's a longer term survival effect. Now, there's a challenge here that this is done with historic controls. And you're actually kind of in this catch-22. Physicians really want to see randomized placebo-controlled studies, but you can't do it anymore with edaravone because it has a significant effect already. So these are some of the things the studies are promising, but there's still kind of a general feeling that this doesn't really work. There are negative studies as well. And so how do you decide to make progress and figure out what to do next? So I'm going to switch now and describe why I got interested in ALS, and hopefully we can swing around and connect these back. So in 1993, there was some mutations discovered in superoxide dismutase, and I had a theory about why this antioxidant enzyme that's protective and found in every cell could be toxic, because we had discovered that it does a bad reaction. You can start putting nitro groups, NO2, onto proteins, and that disrupts their function. So we managed to get a paper published in Nature, which is a pretty big deal. And it was a hypothesis paper. But we'd also developed antibodies that can measure nitrotyrosine, they can detect it. And I went to Harvard, to UC San Francisco, and even to Japan, and found some of the best neuropathologists and gave them our antibodies and talked about how to use them. And they published some really nice papers. So if you look at the bottom, it's shown in black. That was Sam Chu, who actually was here in Portland for his final years. And the black is a signature of motor neurons that are intensely staining for nitrotyrosine. So it was confirmed by multiple people. You could see it in the tissues, and was pretty excited about it. And I want to highlight this, the name Koji Abe, who was the professor in Sendai that I went to visit, because a few years before he was working on stroke, and he had developed edaravone for treating stroke. And that was the initiation where edaravone became used in therapy for stroke in Japan, and also led to his eventual use for treating ALS. There was also, fairly 20 years ago, a study that came out, and they took cerebral spinal fluid and measured nitrotyrosine in the cerebral spinal fluid using these nitrotyrosine antibodies. And what this graph shows is that after 6 months on edaravone, nitrotyrosine basically disappeared. And so I personally found that pretty exciting, because it's saying there's a drug that actually inhibits the signal that we think is responsible for a lot of motor neuron death. But the way it was reported is not terribly convincing. Their methods actually, when you dig back to the paper they refer to, it has nothing to do with measuring nitrotyrosine. And nobody's repeated the work in 20 years, which is usually a sign of people are having trouble repeating it. So we recently returned back to this, and recently is a few years ago, and looking at how does edaravone work? Because one of the challenges is that the way it's given, it only reaches a low concentration in plasma of about less than 10 micromolar at its peak and clears. So how could something that's so low actually work as an antioxidant? What would it do? And so this is where Alex came into the lab and was looking for a project. And so I had him work on in vitro experiments to see how well does edaravone work. So I made my career by something that was very simple. I had this oxidant that we said is produced in vivo. We added it to proteins and they turned yellow. And that's shown as, when you add this oxidant peroxynitrite, you can see the yellow color, I think, in the middle slide. And one of the things, interesting quotes from Isaac Asimov, is that scientific discoveries never start with someone yelling "Eureka." It's always saying, "That's weird." Okay, and I remember very clearly when I saw this yellow color at 11 at night and walking home and saying, "What the heck?" Now we know that uric acid is a good inhibitor, but Alex's moment of, that's weird, is we added peroxynitrite, we added edaravone, and it initially starts being at really low concentrations, a good inhibitor, and then it seems to stop working. And what happens is edaravone itself turns yellow. And so that's what you see here, is that even without it, it's catching the nitro group, that NO2 group, and holding on to it. And that's part of the reason why it works really well. Okay. So I'm summarizing an awful lot of experiments, and I have to thank Alex, because it took a lot of work to get this, what looks like a fairly simple graph out of this, to do it really well. He now works with ALS patients here in Portland and hopes to go to medical school soon. So, is the slide changed? Nope. Can we get the next slide? So I have a graduate student who graduated 20 years ago, and he's ended up being a professor at Emory University, which has a great neural ALS center and ALS center. And Jonathan Glass runs the center, and they did a very large study of collecting cerebral spinal fluid and creating a cohort, and recently published a paper showing that there are a lot of biomarkers that are available. And basically what you're doing is using a really advanced mass spectrometer. They actually have 12 of these machines, and they cost $1 million each. And they take the proteins and digest it, and they mix it with special reagents that allow you to track from which patient did the sample come from. So the bottom line is you can identify a whole lot of biomarkers. And if you look in the plot up on the top, that's called a volcano plot. And two of the biomarkers that the furthest toppest points to the right are neurofilament L and neurofilament M, which are the biomarkers that we've heard about earlier and we'll hear more about. So that was great that you have all these different resources coming together. The technology is like 100,000 times better than when I was working in the lab 20 years ago. So the technical advances of science have been pretty phenomenal. But the interesting thing is that they never bothered to look for nitrotyrosine. It's considered passé. So Blaine Roberts went back and downloaded the data. And Blaine is shown on the bottom with another friend of ours, Peter Crouch in Australia, who we work a lot on ALS. And then the background of the storm clouds gathering over CDC. Which turns out to be pretty pathetic. So if you look at these volcano plots, now we're measuring nitrotyrosine in proteins from ALS patients, and we can identify which proteins. And the way a volcano plot is, it should be two sides, and the ones that are decreased would be on this side, and the ones that are increased would be out here. If you look here, it's more like a plot like this. The nitration's going up. And it turns out you can get nitro groups on tryptophan. It's another amino acid. And we pulled out a whole lot of proteins from there. So you can identify what these proteins are, and it comes to form networks that make a lot of sense. Okay? But you can also look at this as, how well does it differentiate different types of patients? So the Emory group was nicely controlled, and it turns out that there are a lot of different ways of combining the data where we completely differentiate ALS patients from controls. Okay, so now we have actually a really strong set of biomarkers. We're not just taking the proteins, that itself is enormously valuable, but we're looking at oxidative damage on these proteins. Okay, and we can actually go and mine other people's data. The Emory group was just the first, and it's not just ALS. Okay. So we're quite excited about it, and excited that we have the in vitro chemistry to say why this would work. and also these biomarkers that could be used and really adapted fairly quickly. The challenge is that I've gone to physicians and asked, well, can we get some CSF from patients treated with Edaravon? And the answer comes back, oh, it doesn't work. We don't treat our patients with it. So there's this kind of gap in between, and that's why I showed the evidence that some of the, there actually is more evidence with long-term treatment, it works. I'm not claiming that Edaravon is a great drug. It's got a lot of problems, and we can do a whole hell of a lot better. So this is the conclusion that I've got from this, is basically, it's working better than we dared hope, and what people thought initially. It's very clear that Edaravon works to prevent tyrosine nitration, and I would love to talk for many hours or days about how this mechanism works. And Alex has had to escape just to be able to move on with his career. And we have a lot of work over the years that tyrosine nitration is closely linked to the disease, and in fact, it causes motor neurons to die. Okay. So what it means is we can actually make things that would improve the drug. We have a set of biomarkers now to really evolve this and help this go a lot faster. And that's making the assumption that nitration is the target, which for me is a given, but others aren't quite there. The last thing I'd like to say is that today is a day of no kings and so forth. And one of the things that's been under assault is the NIH. And it's hard to say, how much has really evolved. But the ability to do the study that we talked about, that was a $20 million investment that came from indirect costs, and because of the continued support of NIH, and actually the CDC contributed a lot to it. So there is this huge amount of progress that's just really hard to get across in a 5-second soundbite. And we're throwing it away, which is really tragic. So I thank you for your attention, and be happy to talk to anybody afterwards too.
Thank you so much, Dr. Beckman. That was a great presentation. I'm excited to welcome Bets Peters up to the stage. Bets is going to be talking about brain-computer interface. Bets is an ASHA certified speech-language pathologist, and she specializes in augmentative and alternative communication and assistive technology. That's a whole mouthful. She has been with Reclaiming Expressive Knowledge in Elders with Communication Impairments projects since 2012, working with Dr. Melody Fridokin on research aiming to improve communication for people with severe speech and physical impairments. Her research interests include brain-computer interface systems for communication by people with locked-in syndrome, communicative participation for people who use AAC, and improving communication interventions for people with ALS. And it's always wonderful when we get to see you, Bets, and welcome her up. Thank you.
All right. Hi, everybody. I'm excited to be here today. I've attended the symposium a number of times, but I'm happy to be speaking today. I am going to talk about brain-computer interface research, but as well as some other assistive technology-related research that's happening at OHSU. A little bit about me. I am a speech-language pathologist, specializing in augmentative and alternative communication. Basically, augmentative and alternative communication, or AAC, is any kind of communication other than speech, ranging from everything from facial expressions and gestures like all of us maybe use all day, to writing with a pen and paper, to high-tech computer-based speech generating devices. I'm a research assistant professor at OHSU. I serve on the board of the International Brain-Computer Interface Society for researchers and industry representatives in our field. And I used to work as the assistive technology coordinator for ALS Northwest, back when it was the ALS Association many years ago. I also was an AAC specialist at the Portland VA. Now I am mostly doing research at OHSU. So I want to talk a little bit about what BCI is. In our lab, we look at it as an access method for computer control and communication for people with physical impairments. 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 Physical So if you have good hand function, you have a lot of options for controlling a computer. You can use kind of a standard mouse and keyboard, a touch screen, maybe a wheelchair joystick, something like that. If you have control of your eyes, you can use a special camera on a computer to control it with eye movements. Or maybe you can move your head and use a head mouse. Maybe you can move your foot and operate a large trackball that way. Maybe you can twitch your eyebrow and operate a switch that picks up on small muscle activity. But what happens if you don't have any voluntary motor function? This can occur in some cases in a very advanced ALS. It happens in other conditions with people that I work with, brainstem stroke leading to a locked-in syndrome, other neurodegenerative conditions. So how can you control a computer and compose messages to communicate with only your brain without any muscle activity? That is where BCI comes in. It's a method of computer control that does not rely on motor function. You can interact with the environment using only brain signals, and it has a lot of different applications. I focused mostly on communication as a speech-language pathologist, but it is for computer access, controlling a power wheelchair, robotic arms, environmental control, things like that. Often media reports call BCI systems mind-reading machines. That's a phrase that I've heard a lot, and it drives me crazy. These systems do not read your mind. It's not going to, spit out your innermost thoughts without you wanting to and tell your brother-in-law how you really feel about him. It does take intention on the part of the user. So it's really more analogous to controlling a computer with a mouse and keyboard. You're just doing it with your brain instead of with motor function. You're sending a voluntary command to the computer to type out a message or to speak something. So yeah, no mind control. There are a couple different broad categories of BCI. Non-implantable often uses EEG or electroencephalography. That's my dad on the left. I like to experiment on my dad whenever I can. He's wearing a little EEG cap. It's similar to what you wear for a sleep study or something like that. There are also implantable approaches where the user actually has a surgery to implant electrodes on the surface of the brain or within a blood vessel. There's a couple different surgical approaches that can be used for that. At OHSU, we focused on non-implantable EEG-based BCI for communication. We were doing NIH-funded research in that area for 16 years and just ended this summer. Our funding was scheduled to end regardless of what all is happening with the NIH and other funding sources. But I do worry, as we heard in the previous presentation, about what that's going to mean for research going forward. But in these many years, we worked with an interdisciplinary team. We had clinicians from OHSU, so speech-language pathologists, neurologists, EEG specialists. We worked with the electrical and computer engineering team at Northeastern University and natural language processing specialists at Michigan Tech. This was all led by Dr. Melanie Friedoken, who retired earlier this year with the end of our big grant. Our innovations during that time included using language modeling to improve typing accuracy. We created an open source BCI software package called BCI Pi, and that's available for free for researchers or anyone to use. We've investigated new BCI interface designs and typing methods, and we focused a lot recently on customization for individual users. This technology can work really well. Unfortunately, it is more likely to work for people without disabilities in a controlled lab setting than it is to work for the people who could really benefit from it in their homes and communities. So we're focusing on customization. We think that's going to be a piece of making it work better for folks, not just plopping a system down in front of somebody and say, here, use this, one size fits all, everybody gets the same thing. We want to adapt for different people's needs and preferences. Looking at the bigger picture of BCI in 2025, we are seeing an increasing focus on implantable BCI systems. There's a lot of progress being made in this area, both in terms of making the surgery easier and less risky, improving the signal quality, which used to degrade quite a bit over time. That's getting better and better. So they're working on kind of making the surgery more worth doing. They're also making a lot of progress in what those systems can do. There are some implantable systems that are getting close to translating intentional thoughts into speech in nearly real time. This is very, very exciting research. It's still pretty new, but there's some great stuff coming out. I do still think it's important to have non-implantable options, so you can just put on a cap. Not everybody is going to be appropriate to have surgery. Not everybody is going to want to have surgery. So again, I think we just need to have options. We need to have things that can meet people where they're at. There are no commercially available BCI communication systems right now. You can acquire BCI hardware, an electrode cap. You can get BCI software. You can kind of assemble your own system, but there's really nothing that you can get off the shelf that's ready to go for communication and computer access, unfortunately. Certainly nothing that has technical support behind it or insurance funding. With the end of our work at OHSU, there is no active BCI research happening in Oregon right now, but there are BCI study opportunities happening all over the country, and you can search for those on the BCI Society website or using the ALS Registry Research Notification tool. And I'm happy to talk about BCI research or just BCI in general. If you have questions about how it works, what's coming in the future, please feel free to come talk to me in the back afterwards. So I'm not doing BCI research right now, unfortunately, but I do want to talk about some other stuff that I am doing. I'm working with the team on a program called Nomin. This is a web-based typing app. So it doesn't use brain signals, but it is designed for people with minimal movement. So if you can move a finger this much, if you can twitch an eyebrow, if you can move your foot, you can type with Nomin and control a computer with Nomin. We have some research that's going to be starting up soon doing participatory design with Nomin. We want to involve people who are potential users of this app in developing it and refining it and making sure it's actually going to work for folks. So we'll be doing some focus groups, collaborative design sessions, surveys. All of this is done remotely, doesn't involve any travel to a lab or anything like that. We're going to be recruiting soon. We don't have IRB approval yet, so I can't give a whole lot of information, but I am going to have some information about how to contact us so that if you're interested in learning more, when we're able to tell you more, we can follow up with you. And I do have a quick video to show how Nomin works.
Individuals with severe motor impairments, such as cerebral palsy or locked-in syndrome, often communicate using a single switch. Users may control the activation time of their switch by pressing a button, releasing a puff of air, blinking, or some other motion. Special applications take these switch inputs and allow users to communicate or control their computer. Nomon is an interface designed to facilitate fast and flexible selection with a single switch. Shown here is the keyboard application used by participants in our study. Nomon uses clocks to indicate selection options. I first find the letter or word I want to type and look at the clock to its left. Here, I am looking at the H clock.
So H in the middle, second from the top.
I select this
Alphabetical
clock
order.
by activating my switch when the minute hand passes the red noon line. A bell sounds to notify me that I have activated my switch. I repeat this action multiple times until the clock is selected. The screen will flash green to notify me of a selection. I am now selecting the E clock.
An H has appeared in the little window on the left.
The number of switch activations needed to make a select.
We'll move on from that. It's kind of overwhelming to look at it like this. I promise it is pretty easy to learn, and we have a really helpful tutorial on the website that kind of walks you through how it works. So we are going to be looking for some people to participate in research about that, and please come talk to me. Another interesting program that I'm part of now is the AAC Peer Support Network. Our goal is to create a national AAC peer support network. So again, AAC is augmentative and alternative communication, and peer support is just people who use AAC, people who have similar experiences coming together to support each other, to offer and receive help. This can have practical implications for improving AAC use and overall communication. You can learn from how other people are doing things. It can increase feelings of connection, community, and belonging. Can lead to empowerment, feelings of pride, and resilience. And so we're just getting this program started, and we are looking for input from people with communication impairments, from family members, caregivers, healthcare providers. We're looking for people to join this team that's going to be getting this program up and running, and there are opportunities to be compensated for your time. I'm happy to talk more about that opportunity as well. Please come see me in the back. And then finally, another project I've been involved in is related to iPhone and iPad accessibility. There are a lot of settings in iOS that can set up your device for head tracking, for eye tracking, for other alternative access methods. I'm an assistive technology specialist, and I did not know that all of this stuff was there. They keep it really well hidden, and they don't tell anybody about it. It's really hard to find these settings. So I've been working with a guy who had a good friend who unfortunately passed away from ALS. While his friend was alive, he helped him get his iPhone set up for access. He was a software engineer, so this was really his bag. And he was frustrated by how hard it was to do for people who didn't have his technical background. So he created a website with a series of videos that tell you how to do this kind of setup. So I put the address up there at stevesway.com. And the project I've been part of is developing a set of videos and training materials to help speech pathology students and practicing speech pathologists learn how to make these adaptations for people with ALS and other folks who may have difficulty using a touchscreen. So again, we are looking for participants and volunteers for both the NAMAN research and our AAC peer support project. Here are some email addresses you can contact, and I also have some flyers in the back with those addresses, so don't worry if you can't write them down real quickly. And thank you for listening. Do you have a question? What's that? Yeah, that's a fantastic question. So how are BCI researchers thinking about reducing cognitive load and reducing fatigue for using BCI for communication? That is something that is starting to get more attention now. It's something that our lab has focused on quite a bit. A lot of BCI labs are kind of a bunch of engineers in a room. We have really tried to be interdisciplinary with our clinical backgrounds, working with the engineers and trying to bring those things into account. But it is still a huge obstacle. There is a cognitive load. It can be fatiguing. You still have to look at a screen. You don't have the movement aspect, but you still have to focus, you have to pay attention. So we are looking at different adaptations to visual interfaces. Like, for example, the NAMAN interface that I showed you, there's a lot going on there. That's probably not going to be the best for reducing cognitive load. For somebody who really wants to type at a high level, that might be great. A simpler interface might work better for somebody else. So this kind of gets back to the customization aspect that our lab's been working on. We just need to have different options that can work for different folks. All right, thank you very much.
Thank you so much, Bets. All right, and our last speaker this afternoon is Dr. Bob Bowser, and we are excited to welcome him back up after having joined us previously a couple of years ago. So Dr. Bob Bowser is the Chief Scientific Officer of the Chief Scientific Officer of the Denberg Chair for Neuromuscular Disease at Barrow Neurological Institute, where he also serves as the chair of the Department of Translational Neuroscience. He is a leader in ALS research and is known for his groundbreaking work in discovering and validating biomarkers for ALS. And Dr. Bowser also oversees research at Barrow Neurological Institute and St. Joseph's Hospital, co-directs the National Biorepositories of ALS biofluids and postmortem tissues, and participates in numerous clinical trials. Give a warm welcome to Dr. Bowser. Thank you so much for being here.
Thanks, Cassie. And want to first thank ALS Northwest for welcoming me back here. It was, I forget how many years ago now. And I think when I was here before, I was talking about biomarkers, actually a few that Joe was mentioning, things that we had discovered and had been working on for many, many years and sort of translating biomarkers into the clinical arena for ALS. So today I'm going to talk about a different project called All ALS. And this is an NIH-funded National Clinical Research Consortium for ALS. So All ALS actually exists as two national coordinating centers. So I run the Western Coordinating Center, which is at the Barrow Neurological Institute. My colleague, James Berry at Mass General, runs the East Coordinating Center, which is at, again, in Boston at Mass General. So how does this actually function and what does it look like? So in the middle there, you see sort of All ALS again, and All ALS is really Access for All in ALS. It's an acronym we just came up with as we were creating the program. And we work together, completely together, sharing all the protocols, running everything together. We have weekly meetings across our groups. We have 36 participating clinical sites across the country. I oversee 19 of them, and one of those is Dr. Olney's. Yep, there he is. Yeah, Providence is our site here in Oregon. Very happy and proud to have Dr. Olney as a member. And then Mass General, they run 17 other sites. Now above us, you see something called AM ALS. And I don't know if anyone's heard about AMP before. There's actually a couple AMP programs, and first one was in Alzheimer's disease. There's Parkinson's disease. This is ALS, it's probably the third neurodegenerative disease. So AMP stands for Accelerated Medicines Partnership. And this partnership is a public-private partnership. And so it's funded by NIH and funded by the Foundation for NIH, but it also includes pharmaceutical companies, biotechnology companies, foundations that are associated with the disease. And so ALS Association, ALS United, ALS TDI are all members of this partnership that exists. And so there are steering committees that actually are above us, and we have regular meetings with members and leaders within the AMP ALS program. All the money that comes to us from NIH comes from the Act for ALS that we heard a little bit about. And so we're actively pursuing the renewal of the Act for ALS program moving forward to keep a number of national projects, including this one, All ALS, functioning. In addition to what we do, and I'll get into that in a moment, there's also data that's being stored in what's called a knowledge portal that I'll mention at the end. And then all the samples that we're collecting from patients and controls are all being held in a national biorepository at NIH. So what are we trying to do in All ALS? We're really trying to accelerate and advance the future for ALS research and drug development. And so the goals are creating this large and flexible group of clinics around the country to meet both the current but also future research demands. Really creating the sort of the best-in-class type of national consortium that enrolls people throughout the country. So I mentioned there's 36 sites, and I got a picture of the nation with the 36 sites, but we can enroll you wherever you live in the country. So we have fully remote participants, never come to one of our clinics, but they're enrolled in the program. They participate in a number of ways. And every 3 to 4 months, we send a home phlebotomist to their house to collect a blood sample. And so we have a national home phlebotomy group that will go anywhere in the country in order to collect blood samples. We're working with the FDA, so that's part of our partnership too. We actually bring the FDA to some of our other vendors that we're working with within All ALS, developing research study that leads to regulatory approval of their, in this case, mostly devices that are involved in ALS. Again, building this portal to share data. Obviously, accelerating research demands the sharing of data, some of which that Dr. Beckman wishes to have. And so it's the collecting of the information in samples, but then it's sharing everything out. And all the data that gets generated from those samples gets also shared out to the research community worldwide. Plus, we're bringing in all current forms of data into this portal as well. So it'll be a one-stop shopping for investigators. How do we achieve these goals? Again, it's accelerate research, accelerate drug discovery. We got to collect large amounts of data, multiple types of data, and longitudinal clinical information and biofluid samples that connect it all together. We got to integrate and combine that data all together, but then not only for our current studies and to fill the gaps in the prior studies that exist, but then anticipate what's coming in the future. And then again, we have to make all this data and natural history information available to the research community worldwide. Interacting with the community, me being here, Judy in the back has a table around all ALS. So we have a whole outreach program that's part of this effort that is reaching out and going out to various communities in order to both provide information about the program, but also to help our partners locally in those communities to best understand the program that we built, but also to help enrollment of their patients and basically anyone in the population that wishes to participate. And then accessibility. Again, we have 36 sites, but really you can enroll anywhere in this country and participate in all ALS. So within the consortium, within all ALS, we basically have two studies that are ongoing. One that's on the left there is called Assess All ALS. And so these are individuals that have symptomatic ALS or healthy controls. Under this program, we have collection of blood, and you can either opt in or opt out for providing CSF. Again, that frequency is every 4 months, plus or minus a month. And again, the location can be in the clinic or fully remote, or you can start in the clinic and then transfer to a fully remote participant. We actually had one person that started fully remote and wanted to come into the clinic. So we just sort of transitioned them into the clinic. Besides sort of the typical clinical information is collected on the site or fully remote, we also have other endpoints that we're looking at, and digital endpoints that are collected monthly at home by everyone. So even if you're coming to the clinic, there are other activities that you do at home once a month. One is speech. We have one company right now that we're working with, Linus Health, and again, working with the FDA on having regulatory approval for their speech analytics. And then what's called an electronic patient-reported outcome system through another software called NeuroPro, where again, patients are reporting different types of information that's impacting them over time on a monthly basis. The Prevent All ALS on the right, now the participants here are those that are genetically at risk for developing ALS. Setup is about similar, only for these individuals, the clinic visit is only once a year. But again, collecting information and collecting fully remote blood samples on a quarterly basis. And then right now we're starting very soon, it's just rolling out now, another sub-study called Digitize ALS, which will be available for all participants, which is using wearable accelerometers from a company called Empatica. And again, this will be open to both the Assess and Prevent participants. So that's a little bit about the study and what we're trying to do. Again, here's your picture of the nation, of where these sites are located throughout the country. And again, you'll notice the one dot in Oregon where Nick's clinic resides. So we started this, we got funding in late 23. We had to create a program, set up the protocol, set up all the consent forms, get an IRB approval, get all the vendors under contract, et cetera. It was a lot of work. So we didn't enroll the first subject until late 24. And about the beginning, so January of 2025 is when we were sort of fully pushing forward and starting to enroll people across a number of sites, not all of them yet. So and this is numbers as of a couple weeks ago. So as of numbers as of yesterday, we now have 1,010 participants enrolled in all ALS. Thank you. So it is by far the fastest enrollment in an ALS research study that's been performed to date in this country. So we're incredibly excited, but then you see the red bar, and the number goes up and up and up. So we have to enroll another 1,000 within the next 10 months. And so we really need help. And so we really need participation from everyone in this room is eligible. So please feel free to reach out to us and enroll. So it's incredibly exciting. I mean, we work with NIH when they're open. So now they're closed with the government shutdown. So we interact with them on every 2 weeks. We have meetings with leaderships at NIH, leaderships with FNIH, people within the FDA. We have constant communications with them because we have very aggressive enrollment numbers and tremendous amounts of information and data we want to make fully available to researchers around the world. And another great thing, this is our sort of ongoing map of geographic distribution of enrollment. And so we're really looking to have enrollment in every state of the country. We're not quite there yet. So we're at 47 states in Puerto Rico where we have people enrolled. So we're missing a few states, and I think they are Alaska, Hawaii, and I have a lot of volunteers, including Judy, who will happily go to Hawaii and try to recruit people. North Dakota, she didn't volunteer for North Dakota. I don't get that. But we're almost there. Military history, and so this is another important bit of information NIH wanted us to collect moving forward. So right now, about 10%, a little over 10%, are military veterans. So how do you get involved? I think already once a website was shown. So there's a QR code there, there's a website there, there's an email address listed. If you go on a website, some of the information now is old again, because I was saying 989, that was about a week and a half ago. But you'll find information, you can see the latest of the numbers that have been enrolled. On another page, there's a map listing all the sites that are active. If you scroll down from that, it has all the contact information, who directs it, who are their coordinators, how do you contact the coordinators. And if you don't want to do that, maybe you're in North Dakota and you want to participate and we don't have a clinic there. Then on another page, we have something called a participant interest form. We call it a PIF. So you can complete a PIF and submit it, and then we have patient navigators within our program that will reach out to you and help direct you to a site coordinator to lead your enrollment and continued participation in the program. So we have PIF metrics too. So again, picture the country and where PIFs are coming from. And amazingly, I looked at the numbers yesterday again, and so now we're over 400 PIFs that have come to us, and I think we can track off another state that we have at least one from. So we're incredibly happy and excited, but again, a lot of work to do still. And I mentioned this ALS Knowledge Portal. So we're using a company called Sage Bionetworks, and so they have built this portal. It's just going live now. And again, this is where all the information will be housed and made available. So then that's where the magic becomes on how do we accelerate drug discovery and developmental programs throughout the world. Our data will all be here, but not only that, we already have 13 other programs, including Target ALS, Answer ALS, that already are uploading all their information into this portal. And so it'll be a one-stop shopping for everyone. And this is what really helps advance science. As Joe is looking for samples, this is the one place he can go and have access to everything in order to find out where can he get the samples that he really needs for his research. So I think I'm nearing the end. So yes, so we're only successful, because of you and your participation in our programs. So I'm happy to answer questions. And I think the next thing is, it sounds like we have speed dating in the back or something like that, Cassie. So looking forward to it and happy to answer any questions.
Thank you so much, Dr. Bowser. And I love the analogy of speed dating. That is exactly what we're going to be doing next. But before we do that, let's just give another warm round of applause for all of our presenters today. Again, I want to just thank each and every one of you for coming out today. This is one of my favorite days of the year because it's a day that we can really come together, hear from incredibly smart, passionate, and caring people who are doing science, who are doing research, who are providing clinical care to the ALS community. And there's a lot of hope. There's a lot of advancement happening in ALS research, and this day gives me a tremendous amount of hope. So thank you for joining us this afternoon. The day is not done yet. For those who have joined us in the room today, we are going to have a unique opportunity. It is not every day that you can have some individual time with some of these presenters that shared all of this great information with us today. So we'd encourage you to grab another cookie, get some coffee, and go visit all of our speakers at the back of the room to learn a little bit more about how you can get involved as well as answer questions that you have. Once again, thank you to our sponsors, thank you to our speakers, and thank you to each and every one of you for joining us this afternoon. We'll see you at the back of the room.